Everyone. My name is Raisa, Amin, and I'm from the Cellular. My project is about whole animal in vivo screening for small molecule inhibitors of the mitochondrial unfolding response, or the mito-UPR. The mito-UPR is a cellular response that is regulated by transcription of several mitoprotective genes that restore mitochondrial homeostasis. Mito-UPR activation is associated with lifespan extension and protection of hosts during infection. However, recent research has shown that mitochondrial activation makes cancer cells more invasive and metastatic. In fact, mitochondrial target genes like HSP6 and HSP16, which are mitochondrial chaperones, are upregulated in various cancers. ATF5, a mammalian homolog of ATFS1, the key regulator of mito-UPR, is required for cancer survival. Based on these results, we decided to do a chemical screen to look for inhibitors of mito-UPR. Of the 44 chemicals that we found, they had several different functions. They were antimicrobials, neuromodulators, anti-inflammatory agents, etc. Paroxetine, a selective serotonin reuptake inhibitor, works to block serotonin transporters on the presynaptic membrane. This increases the concentration of serotonin in the synapse and boosts emotions of the patients. Paroxetine is used in treatment of depression, anxiety disorders, OCD. We use our HSP6 GFP worms, which are normally used as a readout for mito-UPR, and we put these worms on control paroxetine, bromide, and bromide paroxetine. Under control at paroxetine plates, they had basal levels of fluorescence. In the plate that had the stress bromide, they induce the mito-UPR and were green. However, in presence of bromide and paroxetine, the fluorescence levels were significantly lower, suggesting that paroxetine inhibited the mito-UPR. Next, we did this experiment using HSP4 and HSP16.2 GFP worms. HSP4 is a readout for UPR, and HSP16.2 is used as a readout for cytosolic response. In both cases, the worms with the stressor and the paroxetine had no difference compared to the worms on the plate with the stressor, suggesting that paroxetine only inhibited the mito-UPR. Next, we look into a genetic mutant that induced the mito-UPR called CLOP1. The CLOP1 has a mutation in the ubiquinone, which is a part of the electron transport chain. As a result, it considerably activated the mito-UPR and had a longer lifespan. When the CLOP1 worms were put on the control on the paroxetine plates, we saw that the CLOP1 worms on the paroxetine plates had a significantly lower fluorescence than the ones in the control plate. This suggested that paroxetine inhibited the mito-UPR. When we looked into the lifespan and the survival assay, we saw that the CLOP1 worms on the control had a significantly longer lifespan than the CLOP1 worms on paroxetine. Similarly, when we infected the worms with Pseudomonas aeruginosa, a pathogen, we saw that CLOP1 worms on DMSO had a higher rate of survival than the CLOP1 worms of paroxetine. All of which suggests that paroxetine suppressed the UPRER-associated extension in lifespan and survival during pathogen infection. Next, we look into the clinical target of paroxetine. We know that C. elegans encodes for only one conserved serotonin reuptake transporter, mod5. So we use the mod5 null mutant N3314, and we prepared a cross with our CLOP1 mutant. Using the double mutant, we looked at the fluorescence and the lifespan assay. Both the CLOP1 and the mod5 CLOP1 worms have similar fluorescence levels. Similarly, their lifespans were also similar to each other. This suggests that the clinical target for paroxetine does not inhibit the mito-UPR. Based on all of these results, we are still trying to figure out the exact mechanism through which paroxetine inhibits the mito-UPR. With this, I would like to conclude my presentation. If anyone has any questions, please feel free to email me. Thank you so much for your time.